what was even more striking is if we just looked in the healthy people between younger and older people, there were very prominent changes. So age-related changes in the composition of the blood, and we focused on protein measurements. And that's really the beginning of all this work that we did to try to understand the role of the blood.
Welcome, everybody. This is Longevity Roadmap with Buck Joffrey, and today got a really cool show. I love this one. It reminds me, if you remember this show called Silicon Valley, which is just, I mean, if you haven't seen it, you got to go back and watch it. It was like one of the funniest shows I've seen in a long time, which is basically, it does this thing on Silicon Valley, kind of makes fun of it, like, sort of the oddities of the individuals there and stuff like that. One of the premises was that there was, I guess, one of these billionaires who walked around with his blood donor, like a young man, basically, who basically would do transfusions with to get his young blood in his body. Well, as it turns out, some of that stuff is not just a joke. It's actually kind of real. And so in this show, we're going to talk about, the science behind essentially parabiosis is what that is called. This isn't necessarily entirely about parabiosis, although we do reference it, but it does talk about the real data behind this kind of phenomena of, people having older blood versus younger blood that is diseased with Alzheimer's disease, blood that is from someone who is older, who is completely full intact, and the differences and the potential that creates for therapeutic intervention. So anyway, fascinating show. And we will have that conversation right after these messages.
Hey longevity enthusiasts, it's time to take it to the next level. I've been fine-tuning my longevity regimen for years, and I look better and feel better than I did a decade ago. In fact, my blood work is even better than it was back then. And it's all because of my data-driven regimen, and it's inspired me to create a course and community just for you. It's called the Longevity Roadmap, and I urge you to check it out. If you're tired of your belly fat, tired of being tired, or just want to optimize yourself for the next 50 years, visit longevityroadmap.com. That's longevityroadmap.com.
Welcome back to Longevity Roadmap. Today, my guest is Dr. Tony Wyss-Coray, D.H. Chen Distinguished Professor of Neurology and Neurological Sciences at Stanford and Director of the Phil and Penny Knight Initiative for Brain Resilience. He's one of the world's leading researchers on how systemic factors, particularly molecules circulating in your blood, govern the aging process in the brain. His lab's groundbreaking discovery that young blood can rejuvenate old brains helped redefine aging as a reversible body-wide process, and his translational work has inspired a new generation of potential therapies to preserve cognition and extend health span. Welcome. Tony, how are you?
Thank you. Thanks for having me. I'm good. How are you?
I'm good. This is fascinating stuff. But let's just jump right into it. So your lab's early work showed that old mice exposed to young blood regained cognitive function. So what was the key difference you found between young and old blood that produced these kinds of results?
Yeah, it's a great question. And it really started actually with humans. So I trained as an immunologist and was really interested in understanding the immune system in the brain, and started to work in models for neurodegenerative disease and mostly Alzheimer's disease. And the frustrating part about studying the brain in humans is that we don't really have access to the tissue, right?
Mm-hmm.
We can do cognitive testing, we can do structural measurements, MRI, and so forth, but it's not really possible to get a biopsy except if you get spinal fluid, maybe. That's sort of relatively close to the brain. So we told with the idea that we could potentially get blood samples from patients with Alzheimer's disease and healthy controls and find immune molecules that would differentiate the two groups, or that would even allow us to predict who gets the disease and who doesn't. And the reason why we wanted to do this with focus on the immune system is because people have been showing increasingly in 10, 20 years ago that the immune system seems to have a role in the disease, and there's this inflammation in the brain. So that's actually how we started. It was very difficult at the time because most neurologists, so brain doctors, did not collect blood from patients because they thought this is a brain disease. But we got our hands on a couple hundred samples and did indeed find differences in proteins between healthy people and patients with Alzheimer's disease. But what was even more striking is if we just looked in the healthy people between younger and older people, there were very prominent changes. So age-related changes in the composition of the blood, and we focused on protein measurements. And that's really the beginning of all this work that we did to try to understand the role of the blood in aging, because age is the key risk factor for Alzheimer's disease, and then all these biomarker tests and organ aging and things that we did. So what we found is, again, that the composition of the blood changed between younger people and older people. And the question was then, is this cause or effect?
Mm-hmm. Yeah. So you've turned to, I guess we can define this, but proteomics, right? And tell us what you're kind of looking at there, just so people have an understanding.
Yeah, so initially we focused on immune mediators. These are like hormone-like substances that the immune system uses to communicate with each other. So we call them cytokines, chemokines, interferons, tumor necrosis factor, TNF, which we block with some very well-known drugs to block inflammation in arthritis and things like that. So we measure these types of proteins, but then over the years, more and more proteins became detectable, and we actually use now mostly companies that offer panels of proteins they measure. And they can now detect in just a drop of blood, they can detect thousands of proteins very reliably, reproducibly. And this is really generating sort of a revolution in the field that goes beyond genetics, because in genetics, we could sequence our genome, our DNA blueprint, but now we can measure, on top of that, we can measure proteins very reliably. And these proteins give us more information than just the genes. So this is what's called proteomics.
Yeah, what kind of proteins are they? Are they largely related to the immune system, or are they
go
other
now
types?
way beyond. So one of the companies measures 10,000 proteins, so that's half the genome, if you will. Now, there's some caveats because proteins come in different forms. But even so, we can measure at least one form from half the genome, half the proteins that are encoded in our genome. And so they cover anything from transcription factors to structural proteins, hormone-like proteins, cytokines, immune factors, but really anything. These panels now cover any aspect of biology, really. You can imagine.
So I would imagine that probably machine learning and AI is really probably a big part of this. So essentially trying to, I'm envisioning maybe this is not accurate, but I'm envisioning creating sort of fingerprints, for different expressions. Is
exactly.
Is that right?
Exactly that. So that's what people do now. And most applications use what we call machine learning. So this is not really sophisticated AI, but it basically tries to find. So let's say you have a disease, a person with a disease, you have a person who is healthy, and then you measure 1,000 proteins in all the individuals. And now what the machine learning tools are trying to find is there a protein that is different between the two groups? And it will first rank the one that is the most different, and then it will take the next one and the next one and the next one. And so you can imagine that maybe there's a dozen or sometimes 50 or several hundred proteins that are slightly different. If you take them all together, you get a pretty good model of what makes somebody or describe somebody with the disease compared to a person who is healthy.
I'm trying to tie this together with some of the things we think of, we read about in longevity space right now, specifically, why is it that these protein changes? And the parallel to me when I think about that is, changes to the epigenome, right? Changes to the expression of various genes as we age, which we know it happens in part because of changes to the epigenome. Is that kind of indirectly what we're measuring with the proteomes?
Absolutely. We're basically measuring the expression or the translation of the epigenome. Now, the next question is then, of course, why does the epigenome change? And, most aging researchers think this is really sort of just a result of exposure of an organism to the elements, if you will. And I often compare it to a car, right? You have a car coming off the lot, and they're all built with the same blueprint and should all be the same, but you get sort of over age, the cars will start to break down, and different parts in different cars, even from the same model, even from the same, that were built on the same day. And this is, in a way, it's the result of exposure of that car to the elements, how you drive it, how you treat it, how you service it, but also probably to some extent to randomness, stochasticity, we call it.
Mm-hmm.
And it seems in organisms such as in humans, you have sort of the same thing. You have, with age, everything starts to deteriorate, and we don't think there's a program or there's a purpose in us disappearing, except maybe that evolution needs us to reproduce and go to a next generation to make ever better models of ourselves, if you will.
In terms of brain disease, then, I would think that if you are coming up with a series of fingerprints, then you might, in theory, be able to predict like who's at higher risk of future brain disease. Is that kind of what you're able to do now?
Absolutely, yeah. So that's that's really a very exciting and ever-growing field right now, because of these tools that we can now reliably measure thousands of proteins, but at the same time, there's also sort of healthy aging cohorts, or sometimes they were developed or funded to study heart disease or diabetes or
Sure.
whatever. And so now you have these cohorts with thousands or tens of thousands, sometimes hundreds of thousands of people. Most famously, the UK Biobank, which is 600,000 people that are recruited into these studies. A blood sample is taken from them, their whole medical history, and then they're followed over decades sometimes. The UK Biobank has now 17 years follow-up. So you can now use these types of studies. For example, in the UK Biobank, we have data from 50,000 people 3,000 protein measurements at time zero, and then you have follow-up over 17 years from all of these people. So you can now start to make models, and you can say, okay, what are differences in protein concentrations in people who will later develop Alzheimer's disease, or who will develop, kidney disease or heart disease and so forth? What are proteins to predict how long they live, mortality? And people are, working with this and playing with this, and it's really amazing. And the other really terrific part about this is because we know these proteins, we know the name that makes up these models, right? We discover new biology and potentially new drug targets. So if you find a protein that is a very strong predictor of somebody having, lung cancer, then you might ask, what is the biology of that protein? How does it cause lung cancer? And you can potentially then go into an animal model and see, can I model this? And I think it's really adding a completely new angle to how we study disease, because we can now go from humans to animal models rather than the other way around, how we often have done.
How do you begin to disentangle correlation versus causation? In other words, we know that in somebody with a certain, somebody who's got Alzheimer's disease has a certain fingerprint of proteins. How do you look at that and try to figure out, is it because they have Alzheimer's that they have this fingerprint, or is the fingerprint the causation of the Alzheimer's?
Yeah, that is, of course, key. Now, it's not necessarily that important if you just want to monitor somebody, right? Or if you want to see whether a drug has an effect. A correlate could still be fine. But for drug development, to nominate something as a target, you want to know causation. And the way you can do that, there's different ways. One sort of, not the strongest way, is again to look in people when they're healthy, way before they develop a disease, right? So if something changes very early, it's unlikely going to be just a reflection of a disease process, but it may be more likely a driver, but still no guarantee. What people can do with genetics, if you know the genome of people, you can do something that people call inference, where you use sort of a genetic tool from large population-based genetic studies to see whether, if you then randomize the proteins, whether they might be causative or not. It's a complicated model that people now increasingly use. Mendelian randomization is a term. That gives you even more confidence that something might be causative. But then ultimately, you want to model it either in, cell culture models, animal models, and then finally, you need to test it with a drug that targets whatever you decided might be the cause, and prove that it has an effect on the disease in a clinical trial.
It makes me think, though, in terms of, how you attack this kind of stuff. It makes me think of that show Silicon Valley with when they do, you have this guy, this older billionaire who brings around his young, his blood donor basically to do parabiosis,
right?
He's essentially infusing the younger man's blood into himself. I mean, this has a lot of that flavor. Is there data behind parabiosis in and itself that suggests that might be a legitimate approach to this? Because now you're not dealing with specific targets per se. You're dealing with, just creating a new profile of proteomics for an individual.
Yeah, that's right. Yeah, so in mice, where, we do parabiosis. We don't do it in humans. So parabiosis is really where you join the circulation of two
Yeah.
different animals. And it was actually invented more than 100 years ago. And it led to the discovery of, for example, leptin, this factor that is a mediator of obesity. Sex hormones were described with this because you can, sort of induce sex differences from males to females. But what we've found, and many others in the field now, is that indeed there are factors in the circulation that can change the aging process. So, in other words, as he said earlier, blood from young mice, and specifically the liquid part of the blood, not the cells. We don't need the cells.
The plasma, right?
The plasma,
right?
yeah. If we transfer that from young mice into old, we can rejuvenate many different cell types. And with rejuvenation, we mean they look at a molecular and functional level more like young cells. That's the term people usually use for rejuvenation. And we can do this in very unbiased ways. So we treat the mice and then we take all the cells out and we profile them and we can show they look now younger. But what we also discovered is different cells respond in different ways. Some benefit much more. Stem cells, for example, benefit more from young blood, specifically the stem cells of the immune system. Liver cells also are very responsive, blood vessel cells, endothelial cells, and then others show much less changes. And it may be in part how well these cells have access to the blood, but also the receptors that mediate some of these factors, effects, and so forth. But then the big question is, how does that translate to humans, right?
Yeah. Yeah. Because the idea, in my mind is, well, why can't we just, why can't we do plasma infusions into, older people?
We can.
Yeah.
And that's indeed what we did. So we started a company, Alkahest, a while ago, and worked together with one of the biggest plasma companies in the world, Grifols. They approached this actually from the angle of there are these proteins in Alzheimer's disease that accumulate, called amyloid. And they said, what if we removed the blood from patients with Alzheimer's disease? Would that bring out the amyloid from the brain sort of as a sink? And there were some studies in mice that supported that if you removed the blood and replaced it with, just albumin, which is the main protein in our blood, would that lead to an improvement in these people? And they did indeed find, they had several hundred people that they benefited overall from this treatment. And with our company, with our startup, we then systematically looked at what parts of the plasma might potentially be beneficial. So these companies collect plasma from thousands of donors. They fractionate it into different products that are used in the hospital, that you as a surgeon have used all the time, right?
Yeah, sure.
But also, for example, antibodies, what we call IVIG, is one of the main products that now everybody who has a transplant needs on a regular basis. So we tested these fractions and found some of them are really very beneficial to slow down or reverse this aging process in mice. And so we used then some of these fractions and ran phase 2 trials in Alzheimer's and Parkinson's disease. And that showed they're safe. And we had some positive signs that, people may benefit from this. So the next step is to run large phase 3 trials. And, that requires substantial financial resources. And I hope we're still going in that direction. But that's, that takes some time. But overall, I think the field agrees that there are beneficial factors in young blood that can slow or reverse aging, and there are detrimental factors in old blood. So some people now pursue this idea of what is called plasmapheresis,
Mm-hmm.
and or TPE, and just remove the old factors, because
Yep.
that could also be helpful in itself. And then typically you give back small amounts of albumin to just make up the blood volume. And by the way, that albumin always comes from young donors, because the donor population in these big collection centers is usually younger people. And then also usually they have to give antibodies so that you're not immunodeficient. But there's now companies that offer this actually as a service.
But the albumin-only approach seems to be like you're essentially subtracting, but you're not really adding, right? Because you've got
That's right.
you're
Yeah.
missing you're missing all of those potential beneficial
Yeah.
proteins.
Yeah.
So why would they do that? Why not just supply
Give full
full
plasma.
plasma?
Yeah. Yeah, one could do that too. It's probably a bit more expensive, and in the manufacturing process, that plasma is used for other products. Most importantly, making antibodies. But it's also important to notice that the albumin fraction contains a lot of other factors, and contains actually growth factors, because albumin is the sponge that actually carries a lot of factors around. So even if you just have an albumin infusion, it contains other factors that might be beneficial.
What kinds of other next generation treatments are kind of emerging from this work? Biologics, small molecules, other approaches.
Yeah, what's been a bit frustrating from the research side is to figure out what are the key factors. Because you could imagine, well, what if we just identify what these factors are that are beneficial, and we make them synthetically, and we have a drug, right? But it turns out that it's many different factors. And that makes sense if you think about it. Different cells in our body have different requirements. And it has been very hard to figure out what are the key critical ones, because you need to do this in a living organism. As soon as you take it out into a dish, the cells have different requirements, and we can culture them often, but we can't really test whether that affects aging. Most cells, if you put them in culture, they have a very useful signature. They basically look almost like embryonic cells or like cancer cells, but we can't mimic age in a dish. So we need really these animal models to test aging effects and to test the effects on whole organ physiology.
You focus on the brain for the most part. Obviously, these things are applicable to the entire body, but when you look at what differentiates people who maintain cognitive sharpness late in life versus those who decline early, do you get a sense? I know you're measuring a lot of proteins, but do you have a sense for what kind of patterns or what kinds of things that determine those who have these two different pathways of either not having mental decline versus those who do?
Yeah, sort of resilience, right? Yeah, so we did studies to look specifically at, so one of it was actually with spinal fluid, where we had access to 3,500 people's spinal fluid, and these people had different levels of cognition. And again, we had follow-up over 15 years. So we can look, based on the composition of the spinal fluid, who is at risk to declining cognitively, who is going to be resilient. And we looked specifically independent of the classic pathology of Alzheimer's disease. We were interested in finding something new. And what turned out is that proteins that make up synapses were the best predictors. And we came up with a ratio of just two proteins, which are a very strong predictor whether people are resilient or will decline. Now, you can't go out there and now collect everybody's spinal fluid. That's very invasive.
Yeah.
But we're trying to find similar signatures in the blood. And we found the signatures that, again, was made up with proteins that are involved in making synapses that seem to be a good predictor. Not as strong as the one from the spinal fluid, but I think it's a matter of time until we get this. And so we started a new company, Vero, that is trying to commercialize some of these discoveries that allow us to estimate the age of an organ, the age of your brain, the age of your kidney or heart.
Mm-hmm.
And these are very good predictor whether you will get a disease of that organ. So in other words, if your heart shows to be older than you actually are, you're much more likely to have atrial fibrillation or a heart attack. Same for the brain. And interestingly, the brain age is actually the best predictor for longevity. So those people, out of the 50,000 that we measured also in the UK Biobank, those with the youngest brains live the longest. And those with the youngest immune system and the brain have the best predictor of longevity. People with the oldest brains live the shortest and have a high chance of developing Alzheimer's disease. So this is still research material right now, but we want to translate it to really to a clinical setting in the coming years, and work very closely with clinicians to interpret the findings, right? I mean, there's lots of companies out there that, you can do a test and then it says, okay, you're 5 years older or 5 years younger, and that's it. Nobody tells you what to do with it. So I think what we really need is, first of all, we need detailed information, not about your age in general, but about the age of your organs or the age of your cells, so that we can have specific interventions that are tailored to that organ. And what we already know in standard clinical practice, what would you do if it shows you're at high risk to have heart disease, or high risk to get, lung disease? What would you do? And so working with the clinicians and giving them this new tool that you are at risk because it seems your organ is shows accelerated aging, that hopefully will sort of transform how we really assess health and disease in the future.
When you look at those cohorts, so those different fingerprints that are leading to different outcomes, do you have access to understand what if various lifestyle differences led to those different fingerprints? Is that something that you guys look at?
Yes, absolutely. I mean, there's a bit of a caveat because most of these lifestyle factors are self-reported.
Sure.
So they're not as well controlled, but some are better than others. Like, for example, postmenopausal estrogen replacement. If people report I took estrogen replacement, they probably know. And so there you get actually very good data, I think, where you can show that some of the organs are clearly younger with estrogen replacement.
Interesting.
We also find, people who eat fish, they have younger organs. People who smoke have older organs. Excessive alcohol is associated with older organs. So there is growing data from these, population-based studies. And then I think the next stage is really doing controlled trials where, people have an intervention and you have before and after, and you measure it with these new tools and show that you have now an indicator, first a predictor, but then also a response to your intervention.
You mentioned estrogen. I'm just curious. I mean, did testosterone in men, Did you have that
we
data?
we didn't have that data.
Okay.
It might be in there, but often even in 50,000 people, the numbers might be too small, and then the statistics is not really holding up.
Sure. Well, you now lead this Phil and Penny Knight initiative for brain resilience at Stanford. What's the broader mission of the program, and how does it fit into the future of aging research?
Yeah, that's really an incredible opportunity, and this generous donation that we got. We basically have these funds to support research around trying to understand why some people, live to 90 years or even 100, and their brains are completely intact and they just function perfectly fine.
Mm-hmm.
And then other people, show these early cognitive changes and develop Alzheimer's disease, and really get to understand what are the mechanisms behind that. Support studies at all levels. I mean, it's a Stanford-focused initiative, but we also want to understand just how does the human brain change with age, for example. We don't have really molecular information how the brain changes. So we have large sort of profiling efforts underway where we have people who died. We got their brains from 20 to 100, and we try to understand how does the brain change at a molecular level, and then look at people who were clinically healthy, clinically normal at old age compared to those who had Alzheimer's disease or other degenerative diseases. And the goal is really to build sort of an atlas of how the brain ages and becomes susceptible or resilient, and derive new therapeutic targets that, if you understood why somebody is resilient, what is the key why they didn't get the disease if they're 100 years old? If you could figure that out, you could potentially develop a drug and give this to every one of us, right? That we could all experience this resilience.
Yeah. Fascinating stuff, Tony. I do appreciate you being on the show today. And if people are just curious about the work you're doing or some of these companies, what resources should we turn to?
So the Knight Initiative has a website, and Vero has also a website, Vero Biosciences. Yeah. And just stay tuned on the
absolutely.
the papers that come out. Yeah, thank you so much for your interest. Yeah,
thank you.
Thanks for listening. A quick reminder that while I am, in fact, a surgeon, nothing I say should be construed as medical advice. Now, make sure to include your physician in any medical decisions you make. And also, if you're enjoying the show, please make sure to show your support with a like, share, or subscribe.